In this section, we're going to talk about green fluorescent protein and some other strategies for using genetic engineering in different medical and industrial applications. So first, let's talk about what this protein is. Green fluorescent protein is exactly what it sounds like. It comes from a jellyfish. It's something that jellyfish produce naturally, certain species of jellyfish, and it glows green. The Nobel Prize in, I guess, chemistry was given many years ago for the people who discovered and isolated green fluorescent protein and figured out how to use it in research. So what scientists do is they're able to attach the gene for expressing green fluorescent protein to other genes. So imagine that when you insert a gene into an organism, you're not just inserting one gene, you're actually inserting two that are glued together permanently. And those two genes, one is the target gene expressing whatever, the protein you want, and the other glows green. Well, that's kind of cool. So instead of having to take the DNA, grow it in some way, and then do gel electrophoresis to see if the gene has been taken up, you can just see if it glows green. So you attach it to the gene you want, you allow a bacterium or some other cell, or you insert it into some other cell to take up the gene, and then the cells that glow have the gene you're interested in. So this is one of my favorite ways that people have used green fluorescent protein. The GFP was used in a project with AIDS researchers who are studying resistance to HIV, but they wanted to use cats because cats have a feline immunodeficiency virus, and there is a gene that some cats have that provide immunity to FIV, which is this feline immunodeficiency virus. And they figured if they could figure out how this feline gene produces immunity, then they could use that to produce immunity in humans to the similar HIV virus. And so what they did was they attached green fluorescent protein to the gene that provides resistance to FIV, and then they implanted the gene with both of these proteins, into the eggs of a cat. And then they bred that cat and they examined the kittens. And all they had to do was look at the kittens. Look at the cute little kitty. So here's the mommy cat. She does not have the FIV resistance. Then they've got this adorable little kitten, and they want to see if the kitten has FIV resistance. And so all they need to do is shine a UV light on the cats in the dark, and the kitten glows green. How cute is that? It's a green glowing kitten. No, you may not have one for a pet. What you can have, however, are some other animals, including green glowing fish, because once you've created any kind of green glowing eukaryote, you can create other green glowing eukaryotes. And once you've created green glowing things, you can create things that glow purple and blue and red. So if you want an aquarium full of glowing fish, you can go to Petco and buy those, and they will glow different colors under UV light depending on which genes they have been engineered with. And those are the results of genetic engineering. Other things, researchers have bred rats and pigs with green fluorescent protein in order to study the gene expression in different tissues. So some things that we've learned about how gene expression works were actually learned through these techniques. We can also, for example, attach GFP to another gene for making, say, some kind of antibiotic or special tissues, and by just knowing that the organisms glow green, we know that those organisms will produce the other protein we're interested in as well. So we can use the same kind of thing on a goat. Now we can also do this without the green fluorescent protein. Green fluorescent protein is pretty cool. So anyway, the goat, if you take a gene for producing lysozyme, remember lice? means to break. Lysozyme is an enzyme that can be used as an antibiotic. So you can take this gene and insert it into a goat, breed that goat, and create a population of goats who produce lysozyme in their milk. Then you milk the goats, purify the lysozymes out of the milk, and you've got antibiotics. And so that's how you create a goat that creates antibiotics in its milk. Who would have thunk? And all of these things are the result of not just a lot of scientific thinking and experimentation and repeated failures in experimentation and successes, but also creative thinking. And that's one thing that we don't think about in science, but it's really important that science requires creative thinking, because if you don't have creative thinking, then you can't figure out how to apply these old techniques or these things found in nature to other uses. That requires creativity. So this leads us then to genetically modified foods, which once you've modified some organism, you can modify another eukaryotic organism. By the way, just want to clarify, green fluorescent protein organisms are not for eating. It wouldn't hurt you, as far as I know, to eat something with green fluorescent protein. And I don't know that it would make your tissues glow green, because of course the protein is going to be broken down by your digestive system. So you wouldn't actually ingest into your bloodstream the protein. And the DNA for creating the protein would get broken down into nucleotides, so you wouldn't ingest that and express that gene. You would have to have it injected into you if you were going to glow green. And as far as I know, they haven't figured out how to do anything with that, or no one's done it. Except as tattoos. We have a lot of glowing inks. Not the same thing. Sorry, I should not say that. Not the same thing. Anyway. By adding genes from different organisms to foods, we can create things like a corn that can fight off an insect that eats corn. So the corn makes a protein that kills this insect. That protein is harmless to humans. So now we have a corn that fights off the insect by itself, and farmers don't have to use a pesticide for it. That sounds like a great idea. It's safe to eat. There are economic and environmental impacts of this, and there's a really great video that I also linked to on the Canvas page that talks about some of those issues, and they are serious. However, there is nothing dangerous about eating GM foods. We've been doing it for decades. We're fine. It's fine. There's also a GM rice that makes extra beta carotene. Beta carotene is needed to prevent blindness and some eye diseases. The golden rice that has this extra yellow gene hasn't been used, largely because it was developed without input from the people that it was targeted for, people in parts of the world where rice is a huge part of their diet. And as you might imagine, they were suspicious of it. And then there were other economic impacts as well. We have genetically modified pigs, not with green fluorescent. Again, the green fluorescent protein ones, we don't eat those, okay? But we can still add a different gene to the pigs, and we have, to create pigs that produce more omega-3 fatty acids in their flesh. So this omega-3 fatty acids are normally found in high levels in fish. If you add that gene, add a gene for that fat to pigs, then you get pork that produce more of that. The main benefit of genetic engineering to human health would be in what we call human gene therapy, which is using genetics to treat diseases, both environmental diseases and inherited diseases. Imagine if someone had inherited, for example, a form of blindness, and you could actually fix that, cure that by changing their genes, by inserting the gene that they're not expressing correctly. So that is what gene therapy is. The way this is done is that you take someone who has the normal gene and you insert their gene into a virus. Now, why a virus? A virus is a really common way that is used to insert genes into eukaryotic cells. And that is because the way viruses work, they land on a cell, infect a cell, and they insert their DNA into the nucleus of the eukaryotic cell. And then when the eukaryotic cell expresses genes, it expresses the viral DNA as well. The cell, the eukaryotic cell, actually makes the virus. The virus doesn't make more virus. So by using this mechanism, again, harnessing what nature does naturally, you can insert a gene into the viral DNA, infect the cells with the virus, and that will then insert this viral DNA into the human chromosomes, and the cells will start making the protein on this viral DNA. If you can then get this into bone marrow, then the person's white blood cells and red blood cells will express the proteins. So in this mechanism, the virus is called what we call a vector, meaning that it's the thing that transfers the gene. We normally use this term vector when we're talking about diseases. The thing that transfers a disease is the vector. So we talk about mosquitoes being the vector for malaria or West Nile virus. Ticks are the vector for Lyme disease. Okay, but in this case, we're using viruses as a vector to do genetic engineering. The viral DNA gets inserted into the eukaryotic DNA, and then you take that and insert it into the bone marrow of a person, and now their cells in that bone marrow will express the protein. It has had some successes, and this link is going to take you to a site created by the University of Utah. They have this Genetic Science Learning Center that walks through some of the conditions that have been treated successfully with gene therapy, including hemophilia, certain immune deficiencies, hereditary blindness has been treated by actually getting the brain to respond to visual stimulus. I know, these are amazing. And then certain blood diseases as well, like hemophilia. So that is a nutshell of genetic engineering. I know it's a lot. So hopefully between this lecture and those videos that I've posted on Canvas, this will make sense, and then I'm going to post a review quiz as well. So hopefully you had some fun with that, and next up we're going to look at more genetics.